students, welcome to the PD Life Science Academy for the Happy Learning. NEET एक्झामचे प्रिपरेशन जे विद्यार्थी करत आहेत त्यांच्यासाठी बेसिक कन्सेप्ट लर्निंग आणि लिंकिंग ऑफ कन्सेप्ट हे जेवढे गरजेचे आहे तेवढेच NCERT बुकमधील चॅप्टरचे कंटेंट रीड करणे आवश्यक आहे. हे करत असताना फक्त एक रीडिंग करून चालणार नाही, मल्टिपल टाइम्स रिव्हिजन होणे गरजेचे आहे. यात टाळाटाळ करणे चांगले नव्हे. आपण सेट केलेले टार्गेट अचीव करण्यासाठी PD लाईफ सायन्स अकॅडमीने सर्व स्टुडंटसाठी जे नीट एक्झामचे प्रिपरेशन करीत आहेत त्यांच्यासाठी 21 डे रीडिंग चॅलेंजची सुरुवात केली आहे. रोज एका चॅप्टरचे रीडिंग आपण कंप्लीट करणार आहोत. हे चॅलेंज ज्या विद्यार्थ्यांनी ॲक्सेप्ट केले आहे त्यांनी त्यांचे डेली टार्गेट अचीव केल्यावर कॉमेंट करून सांगायचे आहे. सो लेट्स गेट रेडी फॉर टेकिंग रीडिंग चॅलेंज. ऑल द व्हेरी बेस्ट. थँक्यू.
21 डे बायोलॉजी रीडिंग चॅलेंज. डे 13 रीडिंग चॅलेंज. चॅप्टर नंबर 13 फ्रॉम NCERT बायोलॉजी टेक्स्टबुक ऑफ 11th स्टँडर्ड. चॅप्टर नंबर 13 Photosynthesis in higher plants. All animals including human beings depends on the plant for their food. Have you ever wondered from where the plant get their food? The green plants in fact have to make or rather synthesize the food they need, and all other organisms depends on them for their need. The green plants make or rather synthesize the food they need through the photosynthesis and therefore called as autotrophs. You have already learned that the autotrophic nutrition is found only in a plant, and all other organisms are depend on a green plants for the food are the heterotrophs. The green plant carry out the photosynthesis, a photochemical process by which they use the light energy to drive the synthesis of the organic compound. Ultimately, all living forms on the earth depends on the sunlight for the energy. The use of the energy from sunlight by the plant doing the photosynthesis is the basis of the life on earth. Photosynthesis is important due to two responses, and it is a primary source of the food on the earth. It is also responsible for the release of the oxygen into the atmosphere by the green plants. Have you ever thought what would happen if there were no oxygen to breathe? This chapter focuses on the structure of the photosynthetic machinery and the various reactions that perform or transform the light energy into the chemical energy. Now the 13.1 is what do we know? Let us try to find out what we already know about the photosynthesis. Some simple experiment you may have done in earlier classes have shown in the chlorophyll, that is the green pigment of the leaf, and the light and the CO2 are required for the photosynthesis to occur. You may have carried out the experiment to look for the starch formation in two leaves, the variegated leaves or a leaf that was partially covered with the black paper and exposed to the light. On the testing these leaves for the presence of the starch, it was clear that a photosynthesis occurs only in green parts of the leaves in presence of light. Another experiment you may have carried out where a part of the leaf is enclosed in a test tube containing some KOH soaked cotton which absorb the CO2 while the other half is exposed to air. The setup is then placed in light for some time. On the testing for the presence of the starch later in the two parts of the leaf, you may have found that the exposed part of the leaf tested positive for the starch while the portion that was in the tube tested negative. This shows that the CO2 was required for the photosynthesis. Can you explain how this inclusion could be drawn? Now 13.2 is an early experiment. It is interesting to learn about those simple experiments that leads to the gradual development in our understanding of the photosynthesis. Joseph Priestley in the 1733 and the 1804 In 1770, they performed a series of the experiments that reveals the essential role of the air in the growth of the green plant. Presti, you may recall the discovered the oxygen in the 1774. The Presti observed that the candle burning in a closed space, that is a bell jar, soon get extinguished, and similarly a mouse would soon suffocate in a closed space. He concluded that the burning candle or an animal that breathed the air both somehow damage the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. The Presti hypothesis as follows: the plant restored the air whether the breathing animals and the burning candle removes. Can you imagine how Presti would have conducted an experiment using the candle and the plant? Remember, he would need to rekindle the candle to test whether it burned after few days. How many different ways you can think of the light the candle without disturbing the setup. Using a similar setup as one used by the Presti, but by placing it once in a dark and once in a sunlight. The John Engine House in 1770 to 1799 shows that the sunlight is essential to the plant processes that somehow purify the air fouled by the burning candle or breathing animals. The engine house in an elegant experiment with an aquatic plant shows that in a bright sunlight and some small bubbles were formed around the green parts, while in a dark they do not. Later they identified these bubbles to be of oxygen. Hence the showed that it is only the green part of the plant that could release the oxygen. It was not until about the 1854 that the Julius Van Sach provides the evidences for the production of the glucose when the plant grows. Glucose is usually stored as a starch. His latter study shows that the green substances in the plant, chlorophyll as we know it now, is located in a special bodies that is later called as a chloroplast within a plant cell. He found that the green parts of the plant is where the glucose is made, and that is the glucose is usually stored as a starch. Now consider the interesting experiment done by the T.W. Engelmann in 1843 to the 1909. Using a prism, he splits the light into its spectral component and then illuminate the green alga, that is the cladophora, placed in a suspension of the aerobic bacteria. A bacteria were used to detect the site of the oxygen evolution. He observed that the bacteria accumulated mainly in the region of the blue and the red light of the split spectrum. A first action of the spectrum of the photosynthesis was thus described, and it resembles roughly the absorption spectra for the chlorophyll A and the B. By the middle of the 19th century, the key feature of the plant photosynthesis were known, namely that the plant could use the light energy to make the carbohydrate from the CO2 and the water. The empirical equation representing the total process of the photosynthesis for the oxygen evolving the organism was then understood as the CO2 plus H2O in presence of light is give rise to the CH2O plus O2, where the CH2O represent a carbohydrate, for example the glucose, a 6-carbon sugar. A milestone contribute to the understanding of the photosynthesis was that made by the microbiologist, that is the Carlos van Nijl in 1897 and 1985, who based on his study of the purple and the green bacteria demonstrated that the photosynthesis is essentially a light dependent reaction in which the hydrogen from the suitable oxidizable compound reduces the carbon dioxide to the carbohydrate. This can be expressed by the 2H2A plus CO2 in presence of light. It gives rise to the 2A plus CH2O plus H2O. In green plants, the H2O is a hydrogen donor and is oxidized to the oxygen. Some organisms do not release the oxygen during the photosynthesis, when H2S instead is a hydrogen donor for the purple and the green sulfur bacteria. The oxidation product is a sulfur or a sulfate depending on the organism and not oxygen. Hence, he infers that the O2 evolved by the green plant comes from the H2O, not from the carbon dioxide. And this water latter provides by using the radioisotope technique. The correct equation that would represent the overall process of the photosynthesis is therefore the 6CO2 plus 12H2O in presence of light, that is the C6H12O6 plus 6H2O plus 6O2, where the C6H12O6 represent a glucose. The oxygen release is from the water, and this was proved using the radioisotope technique. Note that this is not a single reaction but a description of the multi-step process called as a photosynthesis. Can you explain why 12 molecules of the water as a substrate are used in the equation given above? Now 13.3 is where does the photosynthesis take place? You would of course answer in the green leaf or in the chloroplast. Based on what you earlier read in chapter 8, you are definitely right. Photosynthesis does not take place in the green leaves of the plant, but it does so also in other green plants of the plant. Can you name some other parts where you think the photosynthesis may occur? You would recollect from the previous unit that the mesophyll cell in the leaves have a large number of the chloroplasts. Usually a chloroplast align themselves along the walls of the mesophyll cell such that they get the optimum quantity of the incident light. When do you think the chloroplast will be aligned with their flat surfaces parallel to the walls? When would they be perpendicular to the incident light? You have studied the structure of the chloroplast in chapter number 8. Within the chloroplast, there is a membranous system consisting of the grana, the stroma lamellae, and the matrix stroma. There is a clear division of the labor within the chloroplast. The membrane system is responsible for the trapping the light energy and also for the synthesis of the ATP and NADPH. In stroma, the enzymatic reaction synthesize the sugar, which in turn form the starch. The former set of the reactions, since they are directly light driven, are called as a light reaction or the photochemical reaction. The latter is not directly light driven by are the dependent on the products of the light reaction, that is the ATP and NADPH. Hence, to distinguish the latter, they are called by the convention as a dark reaction or a carbon reaction. However, this should not be constructed to mean that they occur in a darkness or that they are not a light dependent. Now 13.4 is how many types of the pigments are involved in a photosynthesis. Looking at the plant, have you ever wondered why and how there are so many shades of the green in these leaves and even in a same plant? We can look for the answer to these questions by trying to separate the leaf pigment of any green plant through the paper chromatography. A chromatography separation of the leaf pigment shows that the color that we see in the leaves is not due to the single pigment but due to four pigments, that is the chlorophyll A, which is the bright or a blue green in a chromatogram. Then chlorophyll B is a yellow green, xanthophyll is a yellow, and carotenoid are the yellow to the yellow orange. Let us now see what role various pigments play in a photosynthesis. The pigments are substances that have an ability to absorb the light at specific wavelength. You can guess which is the most abundant plant pigment in the world. Let us study the graph showing the ability of chlorophyll, a pigment, to absorb the light of the different wavelength. Of course, you are familiar with the wavelength of the visible spectrum of the light as well as the Wittgenstein. From figure, you can determine the wavelength, that is the color of the light at which the chlorophyll show a maximum absorption. Does it show another absorption peak at any other wavelength too? If yes, which one? Now look at the figure showing the wavelength at which the maximum photosynthesis occur in plant. Can you see that the wavelength at which there is a maximum absorption by the chlorophyll A, that is in a blue and the red region, also shows the higher rate of the photosynthesis. Hence we can conclude that the chlorophyll A is a chief pigment associated with the photosynthesis. But by looking at the figure, can you say that there is a complete one-to-one overlap between the absorption spectrum of the chlorophyll A and the action spectrum of the photosynthesis? This graph together shows that the most of the photosynthesis take place in a blue and the red region of the spectrum. Some photosynthesis does take place at the other wavelength of the visible spectrum. Let us see how this happens. Though chlorophyll is a major pigment responsible for the trapping light, other thalloid pigments like the chlorophyll B, xanthophyll and carotenoid, which are called the accessory pigment, also absorb the light and transfer the energy to chlorophyll A. Indeed, they not only enable a wider range of the wavelength of incoming light to be utilized for the photosynthesis, but also protect the chlorophyll A from the photooxidation. Now the 13.5 is what is the light reaction. Light reaction or the photochemical phase includes the light absorption, the water splitting, oxygen release and the formation of the high energy chemical intermediate. That is the ATP and NADPH. The several protein complexes are involved in this process. The pigments are organized into the two discrete photochemical light harvesting complexes, that is the LHC, within a photosystem first and photosystem second. These are the name in the sequence their discovery and not the sequences in which they function during the light reaction. The LHC are made up of the hundreds of the pigment molecule bound to the proteins. Each photosystem has all the pigment except one molecule of the chlorophyll A, forming a light harvesting system also called as an antennae. This pigment help to make the photosynthesis more efficient by absorbing the different wavelength of the light. The single chlorophyll A molecule form the reaction center. The reaction center is a different in both the photosystem. In photosystem first, the reaction center chlorophyll A has an absorption peak at the 700 nanometer, hence is called as a P700. While photosystem second, it has an absorption maxima at a 680 nanometer and is called as a P680. Now the 13.6 is the electron transport. The photosystem second reaction center chlorophyll A absorb the 680 nanometer wavelength of the red light, causing the electron to become excited and jump into the orbit further from the atomic nucleus. These electrons are picked up by the electron acceptor, which passes them to an electron transport system consisting of the cytochrome. The movement of the electron is a downhill in terms of the oxidation reduction or a redox potential scale. The electrons are not used up as they pass through the electron transport chain, but are passed on to the pigments of photosystem first. Simultaneously, the electron in the reaction center of the photosystem first are also excited when they receive the red light of the wavelength 700 nanometer and are transferred to another acceptor molecule that has a greater redox potential. These electrons then are moved downhill again, and this time to molecule of the energy rich, the NADP+. The addition of these electron reduces the NADP+ to NADPH plus H+. This whole chain of the transfer of the electron starting from the photosystem second uphill to the acceptor down the electron transport chain to the photosystem first, excitation of the electron, transfer to another acceptor, and finally downhill to the NADP plus, reducing it to the NADP H plus is called as a Z shape due to its characteristic shape. The shape is formed when all the carrier are placed in the sequence on the redox potential scale. Now the 13.6.1 is a splitting of the water. You would then ask, how does the photosystem second supplies the electron continuously? The electron that were moved from the photosystem second must be replaced, and this is achieved by the electron available due to splitting of water. The splitting of water is associated with the photosystem second. The water is split into the two hydrogen and the oxygen and the electrons. This creates the oxygen and one of the net product of the photosynthesis. The electron needed to replace those removed from the photosystem first are provided by the photosystem second. The 2H2O it give rise to the 4H plus O2 plus 4 electron. We need to emphasize here that the water splitting complex is associated with the photosystem second, which itself is physically located on the inner side of the membrane of the thylakoid. Then where are the protons and the oxygen are formed likely to be released? In the lumen or on the outer side of the membrane. Now the 13.6.2 is a cyclic and the non-cyclic photophosphorylation. Living organisms have capability of the extracting the energy from the oxidizable substances and store this in the form of a bond energy. The special substances like ATP carry this energy in their chemical bonds. The process through which the ATP is synthesized by the cell in mitochondria and the chloroplast is named the phosphorylation. The photophosphorylation is the synthesis of the ATP from ADP and the inorganic phosphate in presence of light. When the two photosystems work in the series, the first photosystem second and then the photosystem first, a process is called as a non-cyclic photophosphorylation occurs. The two photosystems are connected through the electron transport chain as seen earlier in the Z shape. Both ATP and NADP H plus are synthesized by the kind of the electron flow. When only photosystem first is a functional, the electron is circulated within the photosystem and the photophosphorylation occurs due to the cyclic flow of electron. A possible location where this could be happening in the stroma lamellae. While the membrane or a lamellae of the grana have both photosystem first and the photosystem second, the stroma lamellae membrane lacks the photosystem second as well as NADP reductase enzyme. The excited electron does not pass on to the NADP plus, but it cycles back to the photosystem first complex through the electron transport chain. The cyclic flow hence results only in the synthesis of ATP but not of the NADP H plus. The cyclic photophosphorylation also occurs when only light of the wavelength beyond the 680 nanometer are available for the excitation. In 13.6.3 is a chemoosmotic hypothesis. Let us now try and understand how actually ATP is synthesized in the chloroplast. The chemoosmotic hypothesis has been put forward to explain the mechanism. Like the respiration in the photosynthesis too, the ATP synthesis is a link to development of the proton gradient across a membrane. And this time these are the membrane of the thylakoid. There is one differences, though here the proton accumulation is towards the inside the membrane, that is in the lumen. In respiration, the proton accumulate in the intermembrane space of the mitochondria when the electron moves through the electron transport chain. Let us understand what causes the proton gradient across the membrane. we need to consider again the process that take place during the activation of the electron and their transport to determine the steps that cause a proton gradient to development. Since the splitting of the water molecule take place on the inner side of the membrane, the process or the hydrogen ion that are produced by the splitting of the water accumulates within the lumen of the thylakoid. B is the electron. An electron move through the photosystem. The protons are transported across the membrane. This happen because the primary acceptor of the electron, which is located towards the outer side of membrane, transfer its electron not to an electron carrier but to the hydrogen carrier. Hence this molecule removes the proton from the stroma while the transporting an electron. When this molecule passes on its electron to the electron carrier on a inner side of the membrane, the proton is released into the inner side or a lumen side of the membrane. Now third is the NADP reductase enzyme is located on the stroma side of the membrane. Along with the electron that comes from the acceptor of the electron of the photosystem first, the proton are necessary for the reduction of the NADP plus to NADPH plus H plus. These protons are also removed from the stroma. Hence within the chloroplast, the proton in a stroma decrease in a number, while in lumen there is an accumulation of the proton. This creates a proton gradient across the thylakoid membrane as well as the measurable decrease in the pH in the lumen. Why are we so interested in the proton gradient? This gradient is important because it is a breakdown of this gradient that leads to the synthesis of ATP. The gradient is broken down due to the movement of the proton across the membrane to the stroma through the transmembrane channel of the CF0 of the ATP synthase. The ATP synthase enzyme consists of the two parts. One called as a CF0 is embedded in the thylakoid membrane and forms the transmembrane channel that carries out the facilitated diffusion of the proton across the membrane. The other portion is called as a CF1 and protrudes on the outer surface of the thylakoid membrane on the side that face the stroma. The breakdown of the gradient provides the enough energy to cause the conformational changes in the CF1 particle at the ATP synthase, which makes the enzyme synthesis several molecules of the energy pack the ATP. The chemoosmosis requires a membrane, a proton pump, a proton gradient and the ATP synthase. The energy is used to pump the proton across the membrane to create a gradient or a high concentration of the proton within a thylakoid lumen. The ATP synthase has a channel that allows the diffusion of the proton back across the membrane, and this releases enough energy to activate the ATP synthase enzyme that catalyzes the formation of ATP along with the NADPH produced by the movement of the electron. The ATP will be used immediately in a biosynthetic reactions taking place in a stroma, the responsible for fixing the CO2 and the synthesis of the sugar. Now 13.7, where are the ATP and NADPH used? We learned that the products of the light energy are the ATP, NADPH and the oxygen. Of these, oxygen diffuse out of the chloroplast, while ATP and NADPH are used to drive the process leading to the synthesis of food more accurately, that is the sugar. And this is the biosynthetic phase of the photosynthesis. The process does not directly depends on the presence of the light, but is depend on the product of the light reaction, that is the ATP and NADPH, beside the CO2 and the water. They may wonder how this could be verified. It is a simple, immediate. After the light becomes the unavailable, the biosynthetic processes continue for some time and then stop. If then the light is made available, the synthesis start again. Can we hence say that calling the biosynthetic phase as a dark reaction is a misnomer? Discuss this amongst yourself. Let us now see how the ATP and NADPH are used in the biosynthetic phases. We saw earlier that the CO2 is combined with the water to produce the glucose or the sugar, and it was interest to scientists to find out how this reaction proceeded, or rather what was the first product formed when a CO2 is taken into the reaction or fixed. Just after World War II, among the several efforts to put radioisotope to beneficial use, and the work of the Melvin Kelvin is exemplary. The use of the radioactivity, that is a C14C isotope by him, a algal photosynthesis study led the discovery that the first CO2 fixation product was the 3-carbon organic compound. He also contributed to working out a complete biosynthetic pathway, and hence is called as a Kelvin cycle after him. The first product identified was a 3-phosphoglyceric acid, and in short the PGA. How many carbons atoms does it have? The scientists also tried to know whether all plants have the PGA as a first product of the CO2 fixation, or whether any other product was formed in other plant. Experiment conducted over a wide range of the plant and leads to the discovery of the another group of the plant where the first stable compound of the CO2 fixation was again and the organic acid, but one of which had a 4 carbon atoms in it. This acid was identified to oxaloacetic acid, or a OAA. Since then the CO2 assimilation during this photosynthesis was said to be 2 main types, and these plants in which the first product of the CO2 fixation is a C3 acid, that is the PGA, that is the C3 pathway, and those in which the first product was a C4 acid, that is the OAA, oxaloacetic acid, that is the C4 pathway. These two groups of the plant shows the other associated characteristics that we will discuss later. Now the 13.7.1 is the primary acceptor of CO2. Let us now ask ourselves a question that was asked by the scientists who were struggling to understand a dark reaction. How many carbon atoms would a molecule have which, after accepting or fixing the CO2, would have the 3 carbons of PGA? The study very unexpectedly shows that the acceptor molecule was a 5 carbon keto sugar, that is the ribulose bisphosphate, that is the RUBP. Did any of you think of this possibility? Do not worry. The scientists also took a long time and conduct a many experiment to reach this conclusion. And they also believe that since the first product was a C3 acid, the primary acceptor would be the 2 carbon compound, and they spent many years trying to identify the 2 carbon compound before they discovered the 5 carbon RUBP. Now the 13.7.2 is a Kelvin cycle. Kelvin and his co-worker then work out the whole pathway and shows that the pathway operated in a cyclic manner. The RUBP was regenerated. Let us now see how the Kelvin pathway operates and where the sugar is synthesized. Let us at the outside understand very clearly that the Kelvin pathway occurs in all photosynthetic plants. It does not matter whether they have the C3 or the C4 or any other pathway. For ease of the understanding, the Kelvin cycle can be described under the 3 stages. First is the carboxylation, second is the reduction, and third is the regeneration. Now first is the carboxylation. Carboxylation is the fixation of CO2 into the stable organic intermediate. The carboxylation is the most crucial step of the Kelvin cycle, where the CO2 is utilized for the carboxylation of the RUBP. This reaction is catalyzed by the enzyme RUBP ADP carboxylase, which result in the formation of the two molecules of the 3-PGA. Since the enzyme also have an the oxygenation activity, it would be more correct to call it RUBP carboxylase oxygenase or Rubisco. Now second is a reduction. These are the series of the reactions that leads to the formation of the glucose. The step involves the utilization of the two molecules of ATP for the phosphorylation and the 2 NADPH for the reduction per CO2 molecule fix. The fixation of the 6 molecule of the CO2 and the 6 turns of the cycle are required for the formation of the 1 molecule of the glucose from this pathway. Now third is a regeneration. Regeneration of the CO2 acceptor molecule, that is the RUBP, is a crucial if the cycle is to continue uninterrupted. The regeneration steps requires the 1 ATP for the phosphorylation to form the RUBP. Hence, for every CO2 molecule entering the Calvin cycle, the 3 molecule of the ATP and the 2 NADPH are required. It is possible to meet these differences in a number of the ATP and NADPH used in the dark reaction that the cyclic phosphorylation take place. To make 1 molecule of the glucose, 6 turns of the cycle are required. Work out how many ATP and NADPH molecules will be required to make 1 molecule of the glucose through the Calvin pathway. It may help you to understand all of these if we look at what goes in and what comes out of the Calvin. Now inside the 6 CO2, 18 ATP and 12 NADPH are required, and there is a out products are the 1 glucose is formed, then 18 ADP plus 12 NADP are formed. Now the 13.8 is the C4 pathway. The plants that are adapted to dry tropical region have the C4 pathway mentioned earlier. Through these plants have a C4 oxaloacetic acid, the first CO2 fixation product. They use the C3 pathway or the Calvin cycle as the main biosynthetic pathway. Then what way are they different from the C3 plants? This question that you may reasonably ask. The C4 plants are the special, and they have the special type of the leaf anatomy, and they tolerate the high temperature. They shows the response to the light intensities, and they lack the process called as a photorespiration and have the greater productivity of the biomass. Let us understand these one by one. The study vertical section of the leaf, one of the C3 plant and the other of the C4 plant. Do you notice the differences? Do both have the same type of the mesophyll? Do they have the similar cells around the vascular bundle sheath? They particularly large cell around the vascular bundles of the C4 plants are called as a bundle sheath cells, and the leaves which have such anatomy is said to be the Kranz anatomy. The Kranz name with a wreaths and is a reflection of the arrangement of the cell. The bundle sheath cell may form the several layers around the vascular bundle, and they are characterized by the having a large number of the chloroplast, thick wall, impervious to gaseous exchange, and no intercellular spaces. You may like to cut a section of the leaves of the C4 plant, maize or a sorghum to observe the Kranz anatomy and the distribution of the mesophyll cell. It would be interesting for you to collect the leaves of the diverse species of the plant around you and cut a vertical section of leaves. The observe under the microscope. Look for the bundle sheath around the vascular bundle. The presence of the bundle sheath would help you identify the C4 plant. Now study the pathway shown in figure. This pathway that has been named the hash and slack pathway is again a cyclic process. Let us study the pathway by listing the steps. The primary CO2 acceptor is a C3 carbon molecule, that is the phosphoenol pyruvate, that is the PEP, and is present in a mesophyll cell. The enzyme responsible for the fixation in the phosphoenol pyruvate carboxylase and the and the pepcase. It is important to register that the mesophyll cell lacks the Rubisco enzyme. The C4 acid, that is the oxaloacetic acid, is a form in the mesophyll cell. It then forms a other C4 carbon compound like the malic acid and aspartic acid in the mesophyll cell itself, which are transported to the bundle sheath cell. In the bundle sheath cell, these C4 acid are broken down to release the CO2 and the 3 carbon molecules. 3 carbon molecule is transported back to the mesophyll, where it is connected to the phosphoenol pyruvate again, and thus completing the cycle. The CO2 released in the bundle sheath cell enters the C3 and the Kelvin's pathways, a pathway common to all plants. The bundle sheath cells are rich in the enzyme ribulose bisphosphate carboxylase oxygenase, that is the Rubisco, but lacks the pepcase. Thus the basic pathway that results in the formation of the sugar, the Kelvin pathway, a common to the C3 and the C4 plant. Did you note that the Kelvin pathway occurs in all mesophyll cell of the C3 plants? If the C4 plant, it does not take place in a mesophyll cell but does so only in a bundle sheath cell. In 13.9 is the photorespiration. Let us try to understand the one more process that creates and the important differences between the C3 and the C4 plant, that is the photorespiration. To understand the photorespiration, we have to know a little bit more about the first step of the Kelvin pathway, the first CO2 fixation step, and this is the reaction where the RUBP combine with the CO2 to form the 2 molecules of the 3-PGA, that is catalyzed by the Rubisco. The RUBP plus CO2 in presence of Rubisco, it give rise to the 3-PGA. The Rubisco, that is the most abundant enzyme in the world, is characterized by the fact that its active site can bind to both CO2 and the O2. Hence the name. The Rubisco has the much greater affinity for the CO2, where when the CO2 as to oxygen is nearly equal. Imagine what would happen if this were not so, and this binding is a comparative. It is the relative concentration of the oxygen and the CO2 that determines which of the two will bind to the enzyme. In the C3 plant, some of the oxygen does bind to the Rubisco, and hence the CO2 fixation is decreased. Here the RUBP, instead of the being converted to the 2 molecule of the PGA, binds with the oxygen to form a 1 molecule of the phosphoglycerate and the phosphoglycolate, that is the 2 carbon, in a pathway called as a photorespiration. In the respiratory pathway, there is neither synthesis of the sugar nor of the ATP. Rather, it results in the release of the CO2 with the utilization of the ATP. In the photorespiratory pathway, there is no synthesis of the ATP or NADPH. The biological function of the photorespiration is not known yet. In C4 plants, the photorespiration does not occur. This is because they have the mechanism that increase the concentration of the CO2 at the enzyme site. This takes place when the C4 acid from the mesophyll is broken down in the bundle sheath cells to release the CO2. This results in the increasing the intracellular concentration of the CO2. In turns, this ensures that the Rubisco function as the carboxylase, minimizing the oxygenase activity. Now that you know that the C4 plant lack the photorespiration, you probably can understand why productivity and yields are better in this plant. In addition, these plants shows the tolerance of the high temperature. Based on the above discussion, can you compare the plant showing the C3 and the C4 pathway? Now next is a 13.10 is a factors affecting the photosynthesis. An understanding of the factors that affects the photosynthesis is necessary. The rate of photosynthesis is a very important in determining the yield of the plant, including the crop plants. The photosynthesis is under the influence of the several factors, both internal, that is the plant, and external factors. The plant factor include the number, size, age and the orientation of the leaf, mesophyll cell and the chloroplast, internal CO2 concentration and the amount of the chlorophyll. The plant or the internal factors are dependent on the genetic predisposition and the growth of the plant. The external factors would include the availability of the sunlight, temperature, CO2 concentration and the water. As the plant photosynthesizes, all these factors will simultaneously affect its rate. Hence, though the several factors interact and simultaneously affect the photosynthesis or the CO2 fixation, usually one factor is a major cause or is the one that limits the rate. Hence, at any point, the rate will be determined by the factor available at the suboptimal level. When several factors affect any biochemical process, the Blackman's in 1905 law of the limiting factor comes into effect. This states the following: if the chemical processes is affected by the more than the one factor, then its rate will be determined by the factor which is the nearest to its the minimal value. It is the factor which directly affects the process if its quantity is a change. For example, despite the presence of the green leaf and the optimal light and the CO2 conditions, the plant may not photosynthesize if the temperature is a very low. And this leaf, if the given the optimal temperature, will start the photosynthesis. In 13.10.1 is a light. First factor is a light. We need to distinguish between the light quantity, light intensity and the duration of exposure to the light while discussing the light as a factor that affects the photosynthesis. There is a linear relationship between the incident light and the CO2 fixation rate at a low light intensity. At higher light intensities, gradually the rate does not show the further increase as other factors become the limiting. What is the interesting to note is that the light saturation occurs at a 10% of the full sunlight. Hence, except for the plant in shades or in dense forest, light is nearly a limiting factor in the nature. Increase in the incident light beyond the point causes the breakdown of the chlorophyll and the decrease in the photosynthesis. In 13.10.2 is a carbon dioxide concentration. Carbon dioxide is a major limiting factor for the photosynthesis. The concentration of the CO2 is a very low in atmosphere between the 0.03 and the 0.04%. Increase in the concentration up to the 0.05% can cause any increase in a CO2 fixation rate. Beyond this, the level can become the damaging over a longer periods. The C3 and the C4 plant respond differently to the CO2 concentration. At low light conditions, neither group respond to the high CO2 conditions. At high light intensity, both the C3 and the C4 plant shows the increase in a rate of the photosynthesis. What is the important to note that the C4 plant shows the saturation at about the 360 microliters per microliters, while the C3 respond to increase the CO2 concentration and the saturation is seen only beyond the 450 microliters per liters. Thus, the current availability of the CO2 level is limiting to the C3 plants. The fact that the C3 plant respond to the higher CO2 concentration by showing the increase the rate of the photosynthesis leading to the higher productivity has been used for the some greenhouse crops such as the tomato and the bell pepper. They are allowed to grow in a carbon dioxide enrich the atmosphere that leads to the higher yield. Now the 13.10.3 is a temperature. The dark reaction being enzymatic are the temperature control. Through the light reaction are also temperature sensitive and they are affected to the much lesser extent. The C4 plant respond to the higher temperature and shows the higher rate of the photosynthesis, while the C3 plant have the much lower temperature optimum. The temperature optimum for the photosynthesis is a different plant also depends on the habitat that they are adapted to. The tropical plant have the higher temperature optimum than the plant adapted to the temperate climate. Now the 13.10.4 is a water. Even though the water is one of the reactant in a light reaction, the effect of the water as a factor is a more through its effect on the plant rather than the directly on the photosynthesis. Water stress cause the stomata to close, hence reducing the CO2 availability. Besides, the water stress also makes the leaves wilt and thus reducing the surface area of the leaves and their metabolic activity as well. Now the summary of the photosynthesis in the higher plant. The green plants make their own food by the photosynthesis. During this process, the carbon dioxide forms the atmosphere is taken in by leaves through the stomata and used for making the carbohydrate, principally the glucose and the starch. The photosynthesis takes place only in the green parts of the plant and mainly the leaves. Within the leaves, the mesophyll cells have the large number of the chloroplasts that are responsible for the CO2 fixation. Within the chloroplast, the membrane are the site for the light reaction, while the chemosynthetic pathways occurs in the stroma. The photosynthesis has two stages, the light reaction and the carbon fixation reaction. In this light reaction, the light energy is absorbed by the pigment present in the antenna and funnel to the special chlorophyll A molecule called as a reaction center chlorophyll. There are the two photosystem first and the photosystem second. Photosystem first has a 700 nanometer absorbing chlorophyll A, that is the P700 molecule at its reaction center, while photosystem second has a P680 reaction center that absorb the red light at a 680 nanometer. After absorbing the light, the electron are excited and transfer through the photosystem second and the photosystem first and finally to the NAD forming NADH. During this process, the proton gradient is created across the membrane to the thallocytes. The broken down of the proton gradient due to the movement through the F0 part of the ATPase enzyme release enough energy for the synthesis of the ATP. The splitting of the water molecule is associated with the photosystem second, resulting in the release of the oxygen, proton and the transfer of electron to the photosystem second. In the carbon fixation cycle, the CO2 is added by the enzyme, that is the Rubisco, to the 5 carbon compound, that is the RUBP, that is converted to the 2 molecule of 3 carbon PGA, and this is then converted to the sugar by Kelvin cycle and RUBP is regenerated. During this process, the ADP and NADH synthesized in a light reaction are utilized, and Rubisco also catalyze the wasteful oxygenation reaction in a C3 plant, that is the photorespiration. Some tropical plant shows the special type of the photosynthesis called as the C4 pathway. In this plant, the first product of the CO2 fixation that takes place in a mesophyll is the C4 carbon compound. In the bundle sheath cells, the Kelvin cycle is carried out for the synthesis of the carbohydrate. Thank you so much.